Welcome to Feedstuffs 365 and National Hog Farmer. I'm Ann Hess. Porcine reproductive and respiratory syndrome caused an estimated $1.2 billion per year in lost production in the U.S. pork industry from 2016 to 2020, an 80% increase from a decade earlier, according to a new analysis by Iowa State University. What can we learn from the latest research in genetics to put into practice? Joining us to discuss this topic and more from the International Conference on Pig Livability is Dr. Jenelle Dunkelberger, Global Health and Behavior Platform Lead with Topigs Norsvin. Welcome and thank you for joining us today.
Thank you so much, Anne. Really appreciate it.
Before we jump into today's conversation, I'd like to remind the audience, if you have any questions for Jenelle, you can post those there in the chat, or if you're tuning in via social media, you can post those there in the comments. So, Jenelle, Topigs Norsvin had a pre-conference here at the Pig Livability Conference. You kicked off your presentation discussing the PRRS Host Genetics Consortium, which found a major gene for PRRS resilience. Give us a little background on this project and your early research in this area.
Yeah, that's right. I mentioned this PHGC project, or PRRS Host Genetics Consortium, which I was a part of as a grad student when I started and did my studies at Iowa State University. That was a really exciting project because it was a collaboration across different groups. So Kansas State was also part of that project in the USDA. It had leaders from those different institutions that were part of that. And it was a multi-year project with this objective of trying to identify a major gene or set of genes associated with enhanced resilience to PRRS. And that was pretty unique and exciting type of consortium at the time, because it was right around that same time that a number of, our breeding company as well as other major breeding companies were starting to implement genetic selection for a number of traits. So it was at this time where we had this whole new methodology or set of tools that we could use to look at the relationship between the host genome and response to various traits. So this was about using that new methodology to see if we could identify regions of the genome associated with just enhanced natural resilience to PRRS challenge.
So you joined Topigs, you talk about the first disease challenge experiment, specifically first looking at a marketing objective. Detail for us what this study was exploring.
Yeah, it was kind of an interesting way that we started in this investigation of looking at genetic variation and disease resilience within our organization. Indeed, coming from more of a marketing objective, because we have a certain terminal line that pigs sired by that terminal line, called the TN tempo. We were getting feedback from customers in different places that they were observing kind of a natural resilience advantage of pigs sired by that line in the face of a disease challenge. And so we wanted to try and investigate that further and see if we could actually quantify that advantage. So we actually designed the trial so that we had a group of pigs sired by that line as well as sired by a competitor line in this side-by-side evaluation. We had those pigs sired by both lines in the barn at the same time, following them from wean to market, and then collecting a lot of really detailed phenotypic information on our animals. Also as kind of it was really a side objective actually to see if what we could find in terms of learning more about the genetic component of disease resilience through this detailed phenotyping. So yeah, originally started as a marketing objective, but kind of saying, okay, we can't miss the opportunity to also collect all this other detailed information on our animals to see if there's an opportunity there to learn more about the genetic basis of disease resilience of that line in particular.
Yeah, let's go into some of those scientific objectives. What were some key findings there?
Yeah, so first of all, in terms of addressing that marketing objective, we did see that there was a highly significant difference in mortality, but growth, and a number of other production traits that we evaluated. So we were able to confirm that kind of intrinsic robustness or resilience advantage. But more exciting actually from a genetic improvement standpoint was the fact that we observed actually tremendous phenotypic and genetic variation even within the temple sired line. So I thought, well, that's really what you need to see and observe to have an indication that you can make further genetic improvement within that line. So for instance, we saw that there were some sires that had as many as 40% progeny losses during the challenge, and then some sires that had 3, 2, or even 0% progeny losses. And that difference that we observed really does have a genetic basis, and we're able to calculate heritability estimates for traits like mortality and growth and clinical scores and treatment.
And you conducted some follow-up studies looking at high resilient progeny versus low resilient. What were some key findings there?
Yeah, that was the next phase of this whole project, is seeing, okay, we observed that this genetic variation exists, so can we actually capitalize on that to estimate breeding values for disease resilience and use that to, in a sense, kind of predict the resilience of progeny coming from these sires. So what we did is we used the data from that initial study to estimate breeding values for disease resilience, or essentially just use the genomic information to predict which sires would have progeny with higher versus lower resilience to disease. Created those progeny, placed them in a barn, put them on test, and subjected them to an experimental challenge trial. And what we saw was that, indeed, the progeny of the boars that we had predicted to have higher resilience did indeed have higher resilience in the face of the challenge, and actually had half as many mortalities as the boars of the low resilient group. So that was pretty encouraging, finding that, okay, we can use just this genetic information in order to make these kind of predictions about response to the disease.
Quite some significant findings there. Through these trials, you also examined heritability, different disease challenges. What did you find in those areas? Is disease resilience a continuous trait?
Good question. Mortality is one of those traits that just under conventional conditions doesn't have a high heritability. We select for it, and I think probably a number of other breeding companies do select for longevity or mortality in some way. But just under conventional conditions, it's not a highly heritable trait. So we were really encouraged to see that actually heritability of mortality under challenge is substantially higher heritability. Of course, any level of heritability indicates that you have an opportunity to improve that trait, but it impacts, of course, the rate of genetic improvement that you're able to achieve. And so with our initial study, we saw that the heritability of mortality was around 10%, which for us was quite encouraging relative to the 2 or 3% heritability under conventional conditions. But then in some of these repeated trials, when we had a better understanding of how to actually execute the experimental design and introducing more genetic variation of the sires represented in the progeny, etc., we were able to achieve, even in one of the trials, a heritability of 31%. So based on the number of trials that we've conducted in between, we have confidence now that heritability under mortality is somewhere between being a lowly and moderately heritable trait. So that's really encouraging from the standpoint of being able to make genetic improvement in this trait.
Yeah, absolutely. Where is Topigs Norsvin going next in this field of research? Can we expect some new disease-resilient products coming out in the market in 2026?
Yeah, thank you for that. We are working towards, let's say, making a new offering to the market in terms of bringing animals with enhanced disease resilience in 2026. That's the plan at this point in time. We've been collecting data to build what we call a reference population, or just a data set of information from challenge trials on an ongoing basis, which has been in place already since 2017. And that's what we need to do. We need to continue to collect information under disease challenge conditions using the most relevant isolates and other diseases that are present, and also, of course, our most recent generation of genetics, because that's constantly changing. But we're going to continue to do that to facilitate continuous updating of these breeding values for disease resilience that we can use as a tool to select and provide animals with enhanced resilience to disease.
You mentioned this overall approach is a natural disease resistance method. For producers at home, what does this mean as far as PRRS control?
Yeah, good question. And maybe just to make a small clarification, we're really focused on resilience rather than resistance, and similar terms, but resilience being more of an umbrella term. So resilience focusing on these animals, they're going to get sick. They're not resistant to viral or bacterial challenges, but they're going to cope with those challenges better, right? So it's not trying to work against the disease, but more working with the disease and just focusing on breeding animals that can continue to grow and thrive despite the challenges that are present.
Dr. Jenelle Dunkelberger, Topigs Norsvin, thank you so much for joining us today and sharing your insight.
Thank you so much for having me. Appreciate it.
For you and our audience, we thank you for tuning in. For more coverage from the International Conference on Pig Livability, be sure to check out the National Hog Farmer and Feedstuffs websites. For Feedstuffs and National Hog Farmer, I'm Ann Hess.